welcome to Erudition Plus. So many of the students asked for crash course in biology, physics and chemistry. So we'll be providing crash courses in bio, physics and chemistry separately. So today we will discuss about biology, and in biology especially human physiology, body fluids and circulation. Body fluids and circulation is basically divided into many parts, but first of all we will discuss vascular connective tissue. So in vascular connective tissue we have divided it in such manner that we have written in which year the question came and what are the most important topics to be covered in vascular connective tissue. So hope you all like the video and understand the concepts behind the video. So this video must be watched after you read NCERT or any textbook which contain minimal information. So here we begin. Vascular connective tissue is divided into blood and lymph. So basically blood and lymph. Blood is red colored, its pH is 7.4, amount of blood in body is 5 liter, while lymph is also a connective tissue which is watery in color, and here I have written it contains more RBC and more WBC and no RBC, and this question came in 2009 AIPMT. So you did one question correct, more WBC and no RBC in lymph. This came in 2009. So now in blood portion, blood is divided into plasma and formed elements. So the plasma is the liquid part of the blood. It contains water and solid part. Plasma is generally straw colored fluid, yellowish in nature. It contains water about 90 to 92%, while the solid part consists of organic substance and inorganic substance. The organic substances are of major use in our exam point of view. So these constitute about 7 to 9% of organic substances. First of all, this is albumin, which is the major component of organic part. Then globulin, fibrinogen, prothrombin, amino acid, glucose and lipids are minor part. So I have written here about globulin. It acts as defense in 2009. How it came in defense? Globulin form antibodies. Antibodies are responsible for defense in our body. Hence globulin for defense came in AIPMT 2009. One question also cleared. Now fibrinogen. Fibrinogen helps in clotting of blood. In consecutive lectures we will clear about fibrinogen. It helps in clotting of blood. This question came in 2011 AIPMT. Another concept cleared. Now prothrombin. Prothrombin is a clotting factor. We will discuss about clotting factor very soon. I hope this portion is cleared well. And the inorganic constituents are not important from exam point of view. This constitute Na+, calcium, K+ and chloride ion, and also bicarbonate ion. Now I will discuss about formed elements. The elements which are formed in blood, they are formed from different parts of the body, especially bone marrow. So in formed elements we will discuss about three major parts: RBC, WBC and platelets. Platelets are also called as thrombocytes. Platelets are formed from megakaryocytes in bone marrow, especially red bone marrow. Now platelets help in clotting. We will know how they will help in clotting very soon. This question came in NEET 2016 that thrombocyte helps in clotting. So I think one more question cleared. Now we will discuss about WBC. WBC, white blood corpuscle. Its count is 6,000 to 8,000 per cubic mm of blood as stated according to NCERT. But the standard value is 4,000 to 11,000 per cubic mm of blood. WBC is basically divided into granulocytes and agranulocytes. In agranulocytes, there are fills. You have to remember fill. See, in granulocyte GP, granulocytes contain all fills, and agranulocytes doesn't contain any fill. They are lymphocyte and monocyte. So granulocyte is eosinophil, basophil, neutrophil. So this is very important according to exam point of view. Eosinophil helps in fights against allergies, worm infestation, etc. While basophil, this question came in 2008 AIPMT. Basophil secrete 5-HT. 5-HT stands for serotonin. Serotonin, vasoactive amine. Hope I I cleared this concept of yours also. Basophil secretes serotonin, histamine also and heparin also, while neutrophil helps in phagocyte. This question came in 2008. Along with neutrophil, phagocytic activity is shown by monocyte also. So granulocytes, what do you mean by granulocyte? For example, these are the cell, here is the nucleus. In the cytoplasm, they contain fine granules. So these are granulocytes, while a granulocyte doesn't contain any granules. Here is lymphocyte, T cell and B cell, they help in immunity. Monocyte is phagocytic in nature. So I hope all the board is clear except one topic which came 2 times in NEET and AIPMT, 2015 and 2007. What do we mean by serum? Serum is basically plasma minus clotting factors. Where is clotting factor in plasma? Here, prothrombin. Prothrombin is clotting factor. So plasma devoid of clotting factor is known as serum. So if we use serum, for example, if we keep serum in room temperature, will it coagulate? No, it will not coagulate just because it lacks clotting factors. Hope I clear this, and now we will turn on to next board. Now we will discuss about RBC. RBC means red blood corpuscle. It contains hemoglobin. It lacks nucleus. It means that it does not have any DNA. It has no DNA. Means mature RBC doesn't contain any DNA or nucleic acid. First of all, RBC was formed in yolk sac when we are in embryo, then in liver, then in bone marrow. Means this question came in AIPMT 2015. RBC formation takes place in many options were given, but the most appropriate option was liver. So hence liver is the answer. Now RBCs were destroyed in spleen. When they were destroyed in spleen, when their lifespan is over, then they were destroyed in spleen. For your information, RBC's lifespan is about 120 days. So spleen is also called as graveyard of RBC. Question came in the pattern, which of the following is known as graveyard of RBC? So the answer will be spleen. This question came in 2012. So mature RBC lacks nucleus. They were formed in yolk sac, liver, bone marrow. Now, for example, I am an adult. My RBC formation takes place in bone marrow, obviously of long bones and skull bones. Hemoglobin content of RBC is 12 to 16 gram per 100 ml. This was asked in AIPMT 2010. I hope this is cleared. Before clearing the board, we will revise once again. Plasma cleared, formed elements, RBC, WBC, platelet cleared, lymph cleared. I hope this time we covered every subject of this board. Thank you. So guys, in this board, we will discuss about blood grouping. First of all, we need to know how frequently blood grouping came in examination, especially in NEET and AIIMS. So in NEET, these type of questions came in 2015, 14, 12, 11th, 9th and so on, if we go backwards. And so we hope this will come in 2019 or upcoming examinations. This will be the favorite question of examiner. In blood grouping, we have to divide blood into different categories according to the presence of antigens. Now, I hope you all know about antigens. Blood grouping system is basically, there are two major blood grouping systems, that is ABO blood grouping system and RH blood grouping system. Blood grouping system was actually discovered by Landsteiner. Now, first of all, we will discuss about ABO blood grouping system and then RH blood grouping system. In ABO blood grouping system, there is presence of antigen on RBC. See, here this is RBC. On the membrane of RBC, there is antigen. See, hope you can see this is antigen. So, there are different antigens present on the membrane of RBC. They can be classified as A antigen or B antigen. First of all, we have to understand what is antigen and how they can show their property. We know that antigen can bind to antibodies. Now, if an antibody is bound to this antigen, obviously RBC will not flow in the blood. RBC will accumulate and they will clump in the blood vessel. So, this will form agglutination of RBCs. So, Hence we know we all are living creatures, our blood is flowing freely. It means that the type of antigen which we have on RBC of the corresponding type of antibody is absent in our blood, hence no agglutination is present in our blood. Now we have to know about genetic factor that consist of ABO blood grouping. There are obviously genes that secrete ABO antigens, AB antigens. So basically it is like we have written IA and IA. This is homozygous, and IA and IO, this is heterozygous. But the phenotypic character will be shown as A blood group. Why? Because O is recessive factor. We have to just know about this only, that O is recessive. Now IA will show A blood group, IO will also show A blood group. But in case of genetic analysis, there will be different results. Now IB will show B blood group, IO will show B blood group. When there is IA and IB, it will show AB blood group, while IO and IO will only show O blood group. Now let us take an example. If a couple married is A blood group and B blood group is crossed, for example they marry, what are the probable offspring that can be possible of different blood groups? So A and B blood group are married. Now they can be, if A blood group can be of IA type or IO type. So what A will secrete? Here one IA or IO. Fine. From B side IO or IB. Now if it is crossed IO, here we will get A blood group, while IB we will fetch AB blood group, and here IO and IO will give us O blood group, while IO and IB will give us B blood group. Hence we can get A blood group children, B blood group children, O blood group and AB blood group. So these are the possible, actually you can say these are the probabilities of getting child as of different blood groups. If A blood group and B blood group individual marries, then these are the following offsprings of A blood group, B, O and AB blood group. Actually this is a crash course video, so we are limited over here only the how to calculate the different kind of offsprings which will be generated. Now ABO blood grouping shows codominance. This is another favorite question of examiner. So ABO blood grouping shows codominance. I hope this is cleared. Now we came to RH blood grouping. RH blood grouping generally derives from RH rhesus monkey, and 85% of Caucasian population, that is we Indians, have RH positive blood group. I have written here positive means presence of antigen, means antigen is present on the RBC membrane. In additional to ABO blood grouping, there is one different antigen known as D antigen. What is known as D antigen is present on the membrane of RBC. That determines whether the individual is positive or negative for RH blood grouping. If it is positive, then it means presence of antigen. Then we can write that blood group as, for example, AB positive. It means A antigen is present, B antigen and D antigen is present on membrane of RBC. I hope this is clear. But negative means absence of antigen. Means, for example, O negative individual have no A antigen, B antigen or D antigen. I hope this is clear. One more important thing is, in RH blood grouping, absence of antigen. It means we have to remember in every blood grouping, if there is absence of antigen, for example, if there is presence of antigen, the corresponding antibodies will be absent. For example, in A blood group individual, A antigen is present. Okay, in A blood group individual, A antigen is present. Then anti-A antibodies will be absent. So what kind of antibodies will be present? Anti-B antibodies. So if we transfuse B blood group into A blood group individual, obviously the RBCs will clump just because the person with A blood group has anti-B antibodies, while, so hope this is clear. Now we'll discuss about something more detailed. in RH grouping, we know that negative means absence of antigen. Here there is absence of antigen, which indicates that there must be presence of antibody, but this is not the case. That's why they are known as incomplete antigen, because here there is no antigen on RBC membrane, but there is also no antibody unless exposed to RH positive blood. Hence, what we can conclude that in an RH negative individual, there is no anti-D antibody. For example, we can take a case of erythroblastosis fetalis over here. I will explain this case to you. First of all, we have to know negative means absence of antigen. Okay, and in RH blood grouping case only, absence of antigen and no antibody unless exposed to RH positive blood. Okay, now let's move on to erythroblastosis fetalis first. In erythroblastosis fetalis, there is RH negative mother. Means, example, we can take any classic example like AB negative mother or B negative mother. For example, B negative mother giving birth to A positive child. So here it is negative and here it is positive. It means that in child there is presence of D antigen. Okay, there is presence of D antigen. While delivery, the blood of fetus interacts with blood of mother. In the blood of, when the antigens go in mother's blood, the mother's blood will form antibodies. What they will form? They will form antibodies against any positive antigen. They will form antibodies. Hence, in consequent delivery, when the mother is again delivering A positive child or any kind of RH positive child, the antibodies will enter into the through the placenta into the fetus and will lyse the RBC. Hope this is clear. So this is known as erythroblastosis fetalis, just because erythroblasts are found in the blood of fetus, because RBCs are lysed. So hence RBC production is overstimulated. When the RBC production is overstimulated, formation of erythroblasts will take place in the bone marrow and in blood. So in peripheral blood smear, we will see erythroblasts in the fetus, and it is a highly risky condition in which the fetus may die just because of hydrops fetalis or something, because when we know that RBC will break down, and end product of RBC breakdown is bilirubin, biliverdin, etc. So bilirubin will accumulate in body and will cause jaundice of the fetus. Hope this is clear. Erythroblastosis fetalis. Basically, we have to know about RH negative mother and RH positive child. That's it. Will cause erythroblastosis fetalis. I hope this blood grouping is clear. Now on the next board, we will go to clotting of blood. Thank you, guys. In this board, we will discuss about coagulation or clotting of blood. Both are generally same. First of all, just in the previous board we missed something. We will recapitulate that. Universal donor is O negative, while universal recipient is AB positive blood group individuals. You can visualize why, as because in universal receptor recipient AB positive, it has A antigen, B antigen, and also the D antigen, that is positive for RH. Hence there is no antibodies in their blood. So any different kind of blood which is given to AB positive individual will not react or not clump. Hence it is clear. While in O negative individual, there is absence of antigen on RBC. Hence RBC given to any other individual can be accepted universally, as because RBC doesn't contain any antigen. So why they will clump with antibody? Hence it is proved that universal donor and universal acceptor fact is cleared. Now in coagulation of blood, first of all, this question came in AIPMT 2010, that which of the ion is required in coagulation or clotting of blood? It is calcium ion. Why calcium ion and how calcium ion? We will see it later on. Now the steps of coagulation. is thromboplastin release. So you will be curious to know about from where the thromboplastin is released. Generally, thromboplastin is released from the platelets when they are injured, or the endothelium, that is the lining of the blood vessel when injured, will release thromboplastin. Now, the thromboplastin which is released will bind with plasma proteins and calcium to activate thrombokinase. When the thrombokinase, this is an enzyme which when it is activated with the help of calcium ion, see here it is the role of calcium ion, thrombokinase will activate prothrombin into thrombin. Prothrombin is a plasma protein and it is a clotting factor, if you do remember from the first slide. With the help of thrombokinase and calcium ion, it is converted into active form, that is thrombin. Now this thrombin helps in conversion of inactive fibrinogen into fibrin threads, and these threads will cover the wound and tighten and helps prevent the blood flowing, and will help in blood clotting. I hope this coagulation is clear, and most of all the question asked will be calcium ion is required, and one more question, which of the vitamin is required in formation of clotting factor. The vitamin is vitamin K. Vitamin K is required for clotting factors, especially 2, 7, this must be written in Roman 2, 7, 9 and 10. For the activation of clotting factor 2, 7, 9, 10, vitamin K is required. Okay. So if your diet is deficient in vitamin K, there will be problem in coagulation pathway, because factor 2, 7, 9, 10 will be inhibited. Only just know about this only, that vitamin K is essential part of coagulation of blood. That's it. Without vitamin K, there will be no clotting. Fine. Now, circulatory pathways. There will be circulatory system, how the blood flows into our body. It is open circulatory system and closed. Closed means from heart. See, if it is heart, the blood is flowing in channels. Means there are different veins and arteries through which the blood is flowing. While in open circulatory system, blood obviously started from heart and some major vessels, and then it opens into the body sinuses. Like it drains into the body sinuses, and then from body sinuses it again goes into the heart. So this is open circulatory system, while this is closed circulatory system. In closed circulatory system, there is a chain in which the blood flows. So closed circulatory system is obviously seen in the most developed animal on this earth, that is we, the mammals, the chordata, the whole chordata family. There are many others in which we can see the closed circulatory system. Now, open circulatory system is seen in arthropods, for example, like cockroaches. Fine, this is clear. I think I have covered the whole board. Now we'll move on to next board. So, next board. So guys, in this video, we will discuss about human circulatory system. First of all, we need to know that human circulatory system is derived from mesoderm. So, human circulatory system is mesodermal in origin. Human circulatory system basically consists of a heart and blood vessels. Heart, which is present between two lungs, slightly tilted towards the left. Hope this is clear. The size of general heart is the size of clenched fist. I hope this is also clear. Now, we will discuss heart is covered externally by double layered pericardium. Inside the pericardium between the two layers, there is pericardial fluid to prevent heart from any injuries. I hope this is clear. Now we will move on to introduction part. This is a free diagram of heart, like we used to draw in physics. Here I also have drawn a free diagram of heart. So basically heart has 4 chambers. We mammals have 4 chambered heart: right atrium, left atrium, right ventricle, left ventricle. So here are the 4 chambers. Now what we have to know is the openings in each of the compartment. First of all, in right atrium always receives deoxygenated or the less oxygenated blood. How? From the coronary sinus, superior vena cava, inferior vena cava. From where superior vena cava carries the blood. Superior vena cava carries blood from our head and neck region, upper limbs and the chest. So from there, the deoxygenated blood is carried out through veins. Veins drain into superior vena cava, and from there, superior vena cava, it reaches the right atrium, while the coronary sinus will drain the deoxygenated blood from the heart itself. So it is the vein of heart, coronary sinus. It will drain into right atrium, while for IVC, means inferior vena cava, it will drain from the rest of the body, that is the lower limbs, abdomen. It will drain into the right atrium. Now, suppose in the right atrium we have deoxygenated blood. Now, where will this deoxygenated blood will go? It will definitely go into right ventricle. But here is an obstruction. There is atrioventricular wall, and there is a gap. Here there is valve is present, known as tricuspid valve. So tricuspid valve is present between right atrium and right ventricle. While now in right ventricle, suppose in right ventricle the blood came, the deoxygenated blood. Now from right ventricle it will go into pulmonary trunk. Here pulmonary trunk is guarded by semilunar valves. I hope this is clear. Now the right ventricle and right atrium, there is separation. Here it is tricuspid valve, means this valve has 3 cusps. I hope you know all about cusp. So it has 3 cusps. Now the 3 cusps are supported by some wire-like structures known as chorda tendineae. See here, cord is coming now. Chorda tendineae. Chorda tendineae supports the cusps of tricuspid valve. So here this question came in 2010. What is the function of chorda tendineae? Chorda tendineae is basically present in the ventricles of both sides and support the cusps of the tricuspid and bicuspid valve both, but mostly found in tricuspid valve because of 3 cusps present in the tricuspid valve. I hope this is clear. Now deoxygenated blood has gone into pulmonary trunk with the help of semilunar valves. Now semilunar pulmonary trunk. Now from pulmonary trunk it will go into the lungs, obviously. Now from the lungs the deoxygenated blood will become oxygenated. Now from the lungs, pulmonary veins will carry the blood. This is PV, means pulmonary veins. Pulmonary veins will carry oxygenated blood. Hence it will contraindicate that veins are carrying oxygenated blood. Hence the modified definition of veins and artery can be illustrated as: artery are those structures that carry blood away from the heart, while veins carry blood towards the heart. Hence this will satisfy all the veins and artery structures of our body. Now oxygenated blood is coming in left atrium. Now from left atrium and left ventricle there is a guarding by bicuspid valve. Hence it has 2 cusps. Now blood came to left ventricle. From left ventricle it will go into aorta, and then aorta through all the body vessels. Now aorta is guarded by semilunar valve. I hope this is clear. Now when the blood is coming in both atria, after atria it will go into ventricle. So closure of tricuspid and bicuspid valve will cause S1. S1 means first heartbeat. First heart sound, actually not beat. First heart sound, S1. And while the closure of pulmonary trunk, semilunar valves of both pulmonary trunk and aorta will produce S2, means second heart sound. So first heart sound is produced by closure of tricuspid and bicuspid valve, while S2 is formed by the closure of semilunar valves. I hope this is clear according to exam point of view, because this is not the detailed illustration of the chapter. Now we will move on to conduction pathway. Conduction, I hope you know, this is the mechanical thing which we are talking about, and now this is the conduction, means conduction of impulses. We know that first of all there is a transmission through nerve and then muscle will contract. So obviously here it is the case. First of all we know that heart is myocardium. There are some cells which became modified and are known as modified myocardial cells. They are SA node, AV node, bundle of His and Purkinje fibers. I hope this is clear. Now I have divided this into 4 chambers so that you can know the location of each nodes and a bundle of His and Purkinje fibers. Now SA node is present in right atrium. In the right atrium, see, upper right part, in the upper right part is SA node. SA node is also known as the pacemaker of the heart because it can generate beat impulse at the rate 70 to 75 per minute. Hence, it is known as pacemaker of the heart. Because of SA node, our heart is creating heartbeats. I hope this is clear. 70 to 75 beats per minute. Now, AV node is also present in the right atrium, the lower left part of the right atrium, and between this is the internodal fibers. Now, the impulse will go from SA node to AV node, then AV node to bundle of His. See, here are two bundles, na? That's why bundle of His. So this question came in 2011. Where are bundle of His present? Option was brain, heart, kidney, lungs, etc. So the answer is heart, obviously. Bundle of His is present in heart. Now, Purkinje fibers. Purkinje fibers are the extension of bundle of His into the each ventricle. So Purkinje fibers also does the work of transmission of impulse in the ventricles. I hope this is clear. Now we will move on to ECG. ECG is only the tracing of conduction of heart, not the mechanical activity of heart. You must know clearly about this. So ECG is graphical representation of electrical activity of heart. This was I was talking about electrical activity of the heart only. Now ECG, this question came in 2013, that is NEET 2013, 11th and 2009 also. So basically this is graphical representation of electrical activity of heart. In ECG, this contains many waves and complexes. Why complexes? Because superimposition of waves will generate complexes. Now P wave, QRS complex and T wave. You just have to know a very little amount of thing. P wave is for depolarization of atrium. QRS complex is for depolarization of ventricles. T wave is for repolarization of ventricles. So where is repolarization of atrium? You must be curious to know about where is the repolarization of atrium. Actually, the QRS complex is so large because of the large mass of ventricle. The repolarization wave of atria is covered under QRS complex. Hence, we can see only QRS complex. Which I mean to say that we can only see repolarization of ventricle, not repolarization of atria. So I think this is clear. Now in heart block, we can assume there is a deformity in ECG. Now we will move on to cardiac cycle. The cyclical event of blood flow, that is cyclical contraction and relaxation of heart, is known as cardiac cycle. Each beat means one cardiac cycle. Each beat takes 0.8 seconds. Hence, in 1 minute, about 72 beats. 72 beats per minute, heartbeat. Fine. Stroke volume is the amount of blood when heart contracts one time, the amount of blood pushed out by each ventricle per beat is known as stroke volume. That is 70 ml. So 70 ml cross 60 will give us 5 liter per minute cardiac output. See, first of all we have to calculate not 60, 70 cross 72. Why 72? Because heart is beating 72 times per minute, and in each beat it is what you can say, it is ejecting out 70 ml of blood. It is ejecting out 70 ml of blood. 70 cross 72 will give us 5 liter per minute. I hope this is clear. I have to be fast because I have to complete the crash course in very limited time. Now heartbeat is 72 per minute. Heartbeat is controlled by parasympathetic system. Parasympathetic system lowers the heartbeat and also the cardiac output, while you can escape the cardiac output part because that must be a very detailed solution. So we have to remember heartbeat 72 per minute. Heartbeat is decreased by parasympathetic, increased by sympathetic. Adrenaline increases the heartbeat, while vagus nerve stimulation can decrease the heartbeat. Fine, this is clear. Now double circulation. You all must be curious to know about double circulation. We humans have double circulation, means same amount of blood goes 2 times through heart is known as double circulation. We have to know only about this. There is a diagram in NCERT where you can easily remember. For example, I can draw over here. This is lungs, This is heart. This is tissue. From heart aorta, it is going to tissues. From tissues, it's coming from veins. From tissue, deoxygenated blood is going from veins to heart, then from hearts to lungs, and then from lungs to heart. So this is double circulation. Basically, you just have to know a certain idea about double circulation. I hope this is clear. Now we will talk about hypertension. Hypertension means increased blood pressure. First of all, we need to know about normal blood pressure. Normal blood pressure is 120/80 mm of Hg. Now, what is 120? 120 is systolic blood pressure, while 80 is diastolic blood pressure. Systolic means when heart contracts, then what is the blood pressure? When a heart relaxes, what is the blood pressure in diastole? Okay, systolic blood pressure is always the highest because it is a contraction of heart. So it is 120, while 80 mm Hg is a diastolic blood pressure. In hypertension, both increases. By then the standard value when we can say hypertension is 140/90 mm Hg. I hope this is clear for hypertension. Now myocardial infarction. In myocardial infarction, the cells of hearts die, that is the myocardium dies. Because of lack of blood supply, there is ischemia because of low oxygen. Hence, in myocardial infarction, we have to treat with streptokinase. This was a NEET question asked in previous years. So in myocardial infarction patient, what you can give is streptokinase, that is clot buster. Here you definitely have read about it in class 12. Now we will move on to two topics only. Blue baby syndrome. Blue baby syndrome is generally increased amount of methemoglobin in blood leads to bluish coloration of skin of newborn, generally because of high amount of methemoglobin in our blood. This is known as blue baby syndrome. While G6PD deficiency, this was asked in previous years of AIPMT around 2005. G6PD deficiency means glucose 6-phosphate dehydrogenase deficiency. Generally, this enzyme prevents the lysis of membrane of RBC. It tries to keep the membrane of RBC intact. How? It destroys the free radicals. It scavenges the free radicals. Because we all know that free radicals destroys RBC's membrane. So G6PD deficiency will always lyse the RBC. Hence, it is clear. I think I have cleared many years question paper along with this crash course. Thank you. If you want to subscribe us, please subscribe us. Share with your friends as much as you can. Thank you.